one of the things I want to kind of get a feel for is we have some tools that other people might like and we might be able to contribute them. But I want to kind of get a feel for whether it's worth doing some of the extra work to make them available more generally. But first, some brief updates of good things that are happening. Cosmic is the external facing sort of component of our group, which is kind of somatic mutations in cancer. Turns 10 next month. I've been using GBrowse for 4 years and we'll be moving to JBrowse on that anniversary. I know some people were interested in potentially seeing integration into other tools, so this is obviously just a simple link to JBrowse itself. With no control from the application. Nothing very special there. However, it's also directly integrated in the aspect that if you go in by gene name, you go straight to the region in the viewer, that specific gene, and you can see all the data. I'm a developer of these tools, so don't ask me too much about the data. But obviously, I'm sure everybody's seen JBrowse and have an idea how it works, etc. And these things are all over the Cosmic website now, so there's tables filled with link outs to JBrowse as well. Interestingly, they still have a G. I assume they're just going with the fact it's a genome browser and they're sticking with that rather than redeveloping all their icons. But they also link out to Ensemble and ChromoView, which is something they've built for doing high-level copy number views. So, moving on from Cosmic, not really my area. Internally within our group, I don't know whether anybody's looked at my poster. I know a few people have, but we have a ridiculous number of BAM files, VCF files, different types of sequencing, species, different builds coming down our pipeline. Our GBrowse server has done an admirable job up to now. Our problem is we can't just make things better by adding image servers, because the problem isn't the number of people using it, it's the amount of data they want to have available at one time. So we've got several thousand data sources, and I've had to patch the fast CGI module to not preload them and stuff like that. But basically it means that even if you haven't preloaded them, when you ask for your data source, it can take 10 seconds for it to find just where it is in the base config file. So it's cricking and falling apart. So when I was moving across JBrowse, I was talking to some of the guys in some of the groups who do use GBrowse, but very minimally. They're going, oh, but you've got to do all the configuration all over again. And I said, well, how would it be if you could have something that was essentially a listing from your database and you give it to a script and it just gave you all the configuration? You might want to tweak a few things through a configuration file. So we've got something that does that. How to contribute it back. It needs a few tweaks, but basically you produce a listing, one line per track, and provides enough information to group that into data sets and projects. You can do, I think it's something in the region of, I think I did 40,000 tracks in about 4 seconds. So it's effectively all you need.
It's faster than Richard did.
Oh, they already leave? Naturally.
Alright.
So all you need is basically a file with, what, depending on how you're doing it, it doesn't have to look remotely like this, other than you would expect to find a result file, potentially an index file, potentially a histogram file in there, depending on the data type. So for us, species and build and project are the important things in the sample. And then obviously you've got result types. But you can control all of this from configuration file of what columns they should group on to create an actual data source. Where's my browser gone? So this is our JBrowse server. We've got a few, and these ones have been grouped on the project column to generate all this. This isn't a big project. This is quite an average one, but 801 tracks straight away, because just for one sample, or a patient, probably is a better way to describe it, you've got a tumour in a normal sample, except that used to be true 4 years ago. We never got more than a tumour in normal, except now we get multi-site sampling of tumours. So we can get 10, 20 samples from one patient.
Do you have any type of control vocabulary so that like, you could see all the ones from the margin of a particular carcinoma, that type of a deal?
So within our, so the Cosmic database within it has a sample management system and a full control vocabulary for describing tissues, phenotypes, all that sort of stuff. It doesn't plug in to grow terms or anything like that, unfortunately. That might change, but it's not my project. We're kind of split up as the teams have grown into bigger groups. But as you can see, the advantage of obviously if you've got somebody who really doesn't want to know how to configure JBrowser, which say, all you need to do is give me a listing of data types and file paths, and it'll do the rest. Would people be interested in something like that? I'm sure somebody will use it.
I think so. I suspect people will.
Yeah.
Okay. I've also been working on something that's, I would be ashamed to show it in here, but basically something that takes that big long listing and allows you to create a completely user configurable version of a configuration. So basically you would sit there and you go, well, yes, obviously I have to change a few things from the same species and build, but I'm going to take 4 samples from this project, 4 samples from this project, and you can inject that into your JBrowser configuration. Now, based on talking to what Rob was talking about yesterday and the changes in JBrowser 2, that's probably not really a major thing to look at because you'll be able to put 2 different data sources on the screen at the same time, so it's not so much of an issue. Okay. So, I will look at that. These are briefly things that I was hoping were going to happen in JBrowser. I've raised a couple of them with Rob already. For us, LDAP authentication, all of the internal systems at Sanger have LDAP access, and it makes authentication single sign-on perfect. And we can control group structure within the institute, so people who aren't in our group don't get access to our servers and things like that. So for us, that's definitely a must. There's a few nice things that we can add to the VCF formats, especially if you've got filters on your VCF data sets. At the moment, it shows the filter abbreviation. You might want a long description available to your users. And something that the first thing I got asked by my scientist, well, what does that mean when it's in that colour? If in the track selector menu, the editor menu, that it would pop, you had the ability to pop up a legend for what your current configuration is, it means you've got to do a lot less documentation for user if it's just pulled out the configuration file. That red is an unpaired read from a BAM file. We also have a variant service. How many people are using VCF as their primary store for data? Are you still using GBrowser as well?
Yeah.
Notice the end at the end, conversion to GFF3 or TSV on the fly. So basically this is a web service written in Java. Your web service can see your VCF files. You do a REST style query against the service. It can either return the data in VCF, a simple tab separated file with a huge header that describes the VCF header as well, or in GFF3. It's a simple GFF3, but it's enough to render components on the screen. You can make minor edits to the configuration, include extra attributes that you could then use callbacks to access. It was basically set up so we could use our VCF files in GBrowse. But I know Ensemble have got an early implementation that they've been playing with. We'd certainly be willing to share it if people are looking to have something like that available. Annotation, standard ontologies. I know everybody likes a standard ontology. So we actually do use sequence ontology terms in an annotation tool. It's been used in Angular within our group for a long time now. About 3-4 years. It works on any species you want it to, provided you can give it some hints as to what the standard, which group is providing the primary gene names. It has been designed so you can use it with any gene or transcript source. So you can use it with Ensemble, or you can use it with UCSC, provided you can tell it how to map that system into its plugin architecture. It also is fast. So when you set it up, you run some scripts which generate bed files which say, here's information about the gene locations, very gross, large information. So basically, if a variant is in your data set which is nowhere near a gene, I stress nowhere near, because we do annotations for upstream and downstream for 3,000 and 5,000 base pairs and possible effects that they could have on some transcripts. It basically will never even attempt to generate an annotation, it'll just say no annotation, because it can just do an intersecting file. And you can then optionally cache information in the very simplistic manner of serialized data into a DB. But 99% of our data no longer actually gets annotated, it pulls it from DB now, because of you running it alone. Any interest? There's an early version already available on the site website if you know the URL, which is a bit useless. It was originally devised for one of the ICGC projects, so that we could have standardized annotation across several groups. Who has very little IT support for their project, especially when it comes to storage infrastructure? Probably nobody. We implemented something before we had iRods on site. The hilarity is track record for things going down. This has never failed, whilst iRods has. Not large scale failed, but you know they've had problems with the databases, or they've had problems with the backup system and file systems. If people want more information on this, by all means get in contact. I'm not going to bore everyone. It works. I think it's this simple way to look at it. So that's all I've got to talk about. Okay. Unless you've got any questions. Thanks. And if anybody wants to talk about high throughput pipelines, that's my other area, lab automation systems. I'm here until tomorrow afternoon.
Okay. All right, let's see. You got 4 words? You want to use your 4 words?
No, honestly, 4 words is really kind of good. I can do it in 2 high